Okay, good morning, everyone. How are you today on this rainy Tuesday? I hope you're doing okay, and I hope you and your family are out of harm's way with the rain from the hurricane coming up the coast here. So, okay, gang, here we are. Congratulations, you made it to your final lecture. Today is it, folks. We got to finish chapter 13 and get some critical information from chapter 14. And Thursday will be your final exam. Before we start, do you have any questions right now in terms of expectations for the end of our semester here, what will happen on Thursday? Quinn, your question, will the final exam have things from all chapters? Not explicitly, no. It will only be the last 4 chapters of your syllabus. But of course, I might expect that you remember things that you've learned in previous chapters. For example, if I talk about a nucleus, you should know what a nucleus is, right? But I will not go out of my way to test you from content of chapter 3. Okay, any other questions? All right, everything looks quiet out there, so I'll assume you're okay. Okay, so, I will reiterate this information in this afternoon's email to you, but again, on Thursday, there will be no lecture because Thursday is the end of the semester. It's the day you take your final exam. So Thursday, there is no lecture, there is no lab. There's simply your responsibility to complete the final exam. Okay? All right, gang. Let's get into it here. Let's continue with chapter 13. And bear with me while I begin to share my screen with you. Okay. So at this point, you should see a Microsoft Word document. I'm reorganizing my screen so the chat window is on the right and the Word document is on the left. Okay. All right, folks. So at the end of last class, we were talking about content from chapter 13, and we had gotten through the second line of defense, right? The title for chapter 13 is Body Defense Mechanisms, and we described the first line of defense, which is your physical and chemical barriers, and the second line of defense, which includes defensive cells, defensive proteins, the inflammatory process, and fever. Okay, I believe that's where we finished at the end of last class. Do you have any questions about those topics before we continue? Okay, not seeing anything. I will continue on to the third line of defense, which is the immune system. Okay. All right. So I wish we had a little bit more time, but I'm going to have to give you some heavy content today. So stay with me and slow me down if you have questions. Okay. Just some opening comments. Okay. The immune system is distinct and unique from the first lines of defense because it represents a specific defense mechanism. It is a specific weapon to defend against a specific invader. Okay. Unlike, okay, typing into your notes, unlike the first and second lines of defense, which are non-specific, okay, non-specific. The immune system is a specific mechanism. Okay, so let's define what we mean by this. By non-specific, when I say that the first and second lines of defense are non-specific, what I'm saying is that these things will defend you in a general way against a variety of potential microorganisms and pathogens, but they will do so in a way where the process really does not know the identity of the microorganism it is defending you against. For example, when your skin prevents something from entering your body, your skin really does not know if it's preventing a bacterium from entering your body, a virus, or a fungal cell. Okay, that makes it non-specific. Okay, think about the room you're in right now. Whatever room you're in, the walls around you are protecting you from the outside world. That wall is kind of a dumb physical object. It does not know that it's protecting you from wind and rain at this moment. It's protecting you, but it doesn't know what it's protecting you from. So the first and second lines of defense are kind of like the dumb wall that's protecting you from the wind and rain outside, okay? The mechanisms at play do not know the identity of the pathogen they are protecting you from. That's what I mean by non-specific. Whereas in a specific response, such as the response of the immune system does know specifically what it is it's protecting you from. The immune system does know the difference between a bacterium and a virus, a fungal cell, that kind of thing. Okay. All right. So stay with me on this. There'll be some philosophical ideas that come out, and hopefully as we wrestle with these philosophical ideas, you'll get a deeper understanding of why things are working the way they are working. Okay, please understand that when I say the immune system knows the identity of the pathogen, I'm not suggesting that your immune system knows the name coronavirus or the name human immunodeficiency virus. The immune system doesn't know these English names that our human brain labels things with. That's not true. But the immune system does know the different chemical makeups of these pathogens and can distinguish them from each other. Okay. All right. So let's see here. We're going to have a series of questions here. Okay. What is the first step your immune system needs to take in order to defend you against a specific threat? Okay. In the chat window, please comment, please answer what you think the answer to that question is. What is the first step your immune system needs to take in order to defend against a specific threat? What is the first step in your immune system? What is the first step your immune system needs to take in order to defend you against a specific threat? What would you say, folks? Okay, Nick and Quinn, I like very much what you were stating there. That's exactly correct. The very first step is recognizing the enemy, identifying what it is that's dangerous to us, okay? Okay, very good. You can't defend yourself if you don't know what it is you're defending yourself from. Okay, Pete, I heard you. I saw your comment there. Okay. Next question. Okay. So if we appreciate that your immune system has to be able to recognize the threat in order to defend you against that threat, the next question is, hold on one moment. The next question is, okay, the next two questions. How does your immune system recognize a threat? What must physically happen? Okay, now let's think about it. In our human experience, if you're going to recognize a threat, let's assume that the threat is another person in the room. Okay, if you're going to recognize a threat, you're going to use your eyeballs and look across the room and identify a person as dangerous for some reason, maybe because they're holding a knife up or something like that. Okay, so you're going to recognize the threat by using your eyeballs. The cells of your immune system do not have eyeballs. So it's not like they can look around your body fluids and say, oh, there's something dangerous. It doesn't work like that. So what would be the analogous response for the cells of the immune system? How do the cells of the immune system recognize the threat? What must physically happen here? Folks, do you have any guesses about this? How do the cells of your immune system recognize the threat since they don't have eyeballs? Okay. Okay, the questions are on the screen. How does your immune system recognize the threat? What must physically happen? All right, so I'm not getting responses, so I'm not sure if you're not understanding or... Okay, Quinn, you say it has something to do with an antigen. All right. Well, I can take that. Okay, so everyone look at Quinn's response in the chat window. She says it has something to do with antigens. Let's take that. So Quinn, how would an immune system cell know that something is an antigen? Okay, Alyssa, you asked, do the antigens have to bump into the cell or attack it? And Quinn, you say they don't chemically match other cells. Well, how do the cells of your body know whether they match or not? Alyssa, I like your answer very much because you say, do the antigens have to bump into the cell? And the answer is yes. That's what I'm fishing for. Okay, if you and I are going to recognize a person as a threat, we're going to use our eyes and look at that person. But cells don't have eyes. So the only way that a cell can recognize a threat is to physically touch it. Okay, what must happen is, that's correct, Alyssa, they're just floating around in there. That's right, they're floating around your body fluids. Okay, what must physically happen is, okay, a defense cell must physically contact a molecule from the pathogen. Okay, a defense cell must physically contact a molecule from the pathogen. if that defense cell does not touch, if it does not touch the molecule from the pathogen, then the defense cell has no hope of recognizing it, no hope of identifying it, and no hope of creating a specific response to fight that threat. Okay? All right. So then does this make sense, folks? That if you want the immune system to recognize an enemy, the cells of the immune system have to be able to touch. They have to be able to bump into, bind with, and touch molecules from the pathogen. Okay, the next question is this. Okay. Let me complete the answer to the second question immediately above with a little bit deeper information. When I say a defense cell must touch a molecule from the pathogen, here's exactly what that is. A defense cell needs to have a receptor that can bind to a molecule from the pathogen. Okay, so we've encountered these ideas of receptors before, right? We talked about insulin receptors in the endocrine system. Well, the receptor here is not binding a hormone, but it would bind something else specifically from the pathogen. So a defense cell must have a receptor capable of binding a molecule from the pathogen. That has to happen in order for the immune system to recognize the threat. So my next question to you is this. How does the immune system recognize a pathogen it has never seen before? Okay, so, forgive me for repeating myself, but sometimes I feel it's important to say things different ways so that you really get what we're trying to have you understand. How does the immune system recognize a pathogen it has never seen before? If recognition requires that a defense cell have a receptor capable of binding that pathogen, how in the world can I have a defense cell with that receptor if the body's never seen that pathogen before? So my question is, how does the immune system recognize a pathogen it has never seen before? Folks, how does that work? Okay, Nick, you say it has to make new antibodies. I appreciate the comment. There's no way that the immune system will make antibodies if you don't have a binding event first. There must be a binding event, which is this event up here of a defense cell with a receptor binding to a molecule from the pathogen. That must happen before you can generate antibodies. Alyssa, you say you don't think that the immune system recognizes it. Isn't that why we have vaccines? Well, Alyssa, you are correct that a vaccine will expose you to something and give you that immunity. But please understand that 200 years ago, there were people that were alive and their immune systems still defended them against threats in the environment, and there were no vaccines around. So what I'm saying is that the immune system can recognize something it's never seen before. So how does that work? Okay, Pete, I'm sorry you're having technical problems. Do the best you can. All right. Okay, folks. So I'm going to momentarily stop sharing my screen so I can see you face to face so we can talk about this. Okay. Okay. If you're able to turn your video camera on, it's much appreciated. Okay. Your immune system is capable of recognizing a pathogen the body's never encountered before. How in the world does this happen? Okay, let me ask you an alternative question for an analogy to this concept. All right, tomorrow is Wednesday, and if I'm not wrong, usually Wednesday is one of the days of the week that the Powerball lottery is drawn, where if you've purchased a ticket, you could potentially win the Powerball lottery. Okay? Let me ask you, how could you guarantee that you will win the Powerball lottery tomorrow without cheating? How could you guarantee that you will win the Powerball lottery tomorrow without cheating? Okay. All right. Julia, Quinn, Nick. Okay. Beautiful, Matt. In other words, buy all the tickets with every conceivable number combination, and you will definitely win the Powerball lottery tomorrow. Now, from a pragmatic human experience perspective, that's not a good strategy because you would have to spend more money buying all the tickets than the amount of money you would win in the lottery itself, right? So that's not useful from that perspective, right? But you see that mathematically, it is possible to guarantee yourself that you will win the lottery by buying all the tickets with every conceivable number combination. Before we go any further, does that idea make sense to you? Does anybody need me to explain it differently? Okay, it's like this. Now, I've already given you your study guide for the final exam, but let's pretend for a moment that I did not give you a study guide for the final exam. If I had not given you a study guide for the final exam, then your options would be to give up or basically to study everything, correct? In other words, prepare for everything. So that's the idea that the immune system takes. The immune system goes about building defense cells, and every defense cell is kind of like a lottery ticket where it doesn't have a unique number combination to win the lottery, but it does have a unique receptor shape to win the lottery, which is the binding to the pathogen. Okay, let me say that one more time. Your immune system prepares for future infections against pathogens it has never seen before by building an army of defense cells and placing randomly shaped receptors on their surface, with the expectation that one of these randomly shaped receptors will have the correct shape to bind any molecule from any future pathogen that you might be unfortunate enough to become infected with. Does this idea make sense? Okay. All right, folks, I'm gonna codify that in your notes at this time, and we're going to continue. Okay. All right, let's continue here. All right. So the answer to this question is your immune system is constantly manufacturing new defense cells and giving each cell a randomly shaped receptor potentially capable of binding any foreign molecule that enters your body. Okay? Now, you might be sitting there thinking to yourself, well, surely your immune system cannot build an infinite number of receptors, and you would be correct. But it can build a very large variety of different receptors. Okay, current estimates from science suggests that your body can build at least 10 to the 15th power different receptors. Okay? Current estimates from science suggest that your body and the genes in your cells can build at least 10 to the 15th different receptors. That's a very, very large number of different things. Okay? All right. Gang, any questions at this time? Okay. All right. Okay. All right. So the way that your body builds all these different receptors is it takes your DNA and it kind of shuffles different sequences of DNA together in different arrangements, kind of like shuffling a deck of playing cards. Okay. So your body builds all these different receptors by shuffling segments of DNA, genes, inside your immune cells as though they were cards in a deck of playing cards. Okay, so here's another question for you. Folks, help me out here because sometimes my old brain doesn't remember things. How many cards are there in a standard deck of playing cards? Okay, Matt, thank you. 52. All right, so here's my question. Thanks, everybody. How many different ways can you shuffle, meaning arrange all 52 cards in a deck of playing cards? Okay, you ever think about that? Maybe you have and you would never admit it. Or maybe you didn't because it's just too geeky of a question. Either way, we got to think about it now. How many different ways can you shuffle, meaning arrange, all 52 cards in a deck of playing cards? How many unique ways could you arrange all 52 cards after any given shuffle? How many unique ways can that be done? Can you calculate? Do you know how to calculate that? Okay, Matt, you gonna shuffle them all out? Gotcha. I appreciate that. Okay. It's not 52, Catherine. This is mathematically solvable, and that's why I typically challenge students with it. I appreciate that some of you may not have had this math, or you may have had this math a very long time ago and you kind of forget it. Okay. All right. So, okay. So, folks, can anybody tell me what the mathematical operation is called that you would push a button for on your calculator to answer this? Matt, a summation could be part of the answer. I would agree with you on that. It would not be the entire answer. Depends how you set it up. Okay. So, so as not to get bogged down here, folks, what I'm fishing for is this. You would do 52 factorial, which on your calculator, if you have a calculator available, is 52 with an exclamation point. You're onto the right concept, Matt. You're onto the right concept. Okay, let's do that together. Let me pull up the old calculator here on the shared screen and do that for you. Okay, so okay, so right here, if you can see my window, there's the n with an exclamation point. That's factorial. Okay, what that's going to do is answer our question for us. So if you have a calculator and you push that button, the computer is programmed to solve a little mathematical formula that will answer questions like this. Good job, Matt. I think that's right. 52 factorial, 8.06 times 10 to the 67th power. That's exactly right. Well done. In other words, there are 8 times 10 to the 67th power unique ways of shuffling all 52 cards in a deck of playing cards. Okay? All right? That's the answer to the question. Now that seems absurd, but here's what I'm doing. Imagine instead of having 52 cards, we have 52 segments of DNA, 52 genes, if you will, and we shuffle them around in different combinations, different arrangements, if you will. You can see by shuffling around the DNA as though they're cards in a deck of cards, we can build an incredibly large library of different molecules. Certainly larger than 10 to the 15th power. Okay, so I'm not going to take this concept any further, but I just want to give you an idea of how your cells do this. Your cells literally rearrange the segments of DNA by shuffling them around like they're cards in a deck of playing cards. And by doing this, you can build a phenomenally large library or army of cells with unique receptors capable of binding things that your body has never seen before. Okay? That is pretty powerful, wouldn't you say so? Okay, do you have any questions right now? All right. So let's do this. I'm going to introduce you to a couple cells that you need to be familiar with to understand the immune system response. Then I'm going to show you a little video to give you some visualization on these ideas, because I appreciate that if you've never studied this before, having a foundation, a graphical cartoon to appreciate things might allow you to understand the lecture easier. Okay? Okay. Cells involved in the immune system. Okay? The immune system is a powerful adaptive weapon to fight dangerous threats to you folks. It is so powerful because it can adapt. It can adapt to a unique pathogen. All right? Cells involved in the immune response. Okay, this will not be an exhaustive list, but just a couple things so that you're aware of what we're talking about. What I'm talking about are B lymphocytes and T lymphocytes. Okay, these are your primary warriors of the adaptive immune system. Okay, your B lymphocytes are born in the red bone marrow. Okay, they're manufactured, they are reproduced from stem cells in the red bone marrow. And so are the T cells. They are also born in the red bone marrow. Okay. Now, after each type of lymphocyte is manufactured in the bone marrow, they also go through a period of maturation, or you might say a period of training, like you folks are in school right now, so you're training to know more about biology, and that may support your future career, whatever you decide to do. Okay, so the B lymphocytes are born in the red bone marrow, and they remain in the bone marrow for training and maturation. Okay, now where this differs is the T lymphocytes. The T lymphocytes, they physically migrate to your thymus. They go to the thymus for their training and maturation. So you can think T lymphocyte, T for thymus. All right. So, any questions right now? Okay. Folks, let's watch a little cartoon here that's just going to open, hopefully, a different way of thinking about things. And then we're going to step through an immune response in a way that will help you prepare for your final exam, okay? So hold on here. I'm going to go to YouTube and show you this video. If for some reason you can't see it, definitely speak up with your microphone so that I can fix that. Okay, here we go. All right. Here's a video, the innate and adaptive immune systems by people associated with the Vaccine Makers Project. Here we go.
The immune system's first defense is our skin, the largest organ of our body. The skin covers a total area of about 2 square meters, the size of a typical basketball backboard. When microbes attack, they're routinely deflected by the skin. Inside our bodies, mucous membranes are slimy, warm surfaces that make up about 400 square meters, roughly the size of an entire basketball court. They're under constant attack from microbes in the air we breathe and the food we eat. They line our nose, throat, intestines, and reproductive tracts. That's a large area to defend. Your mucous membranes are going to need help. Fortunately, certain cells in your body have evolved to fend off microscopic attackers. Early responders, like this macrophage, identify invaders and attack. They also give off chemical signals, called cytokines, to call up reinforcements like neutrophils and natural killer cells. Macrophages, neutrophils, and natural killer cells, collectively referred to as innate immunity, are nonspecific, often sacrificing healthy tissue to contain an infection. But if the invader slips through, you're going to need a more targeted defense. The adaptive immune system provides a more targeted defense. Dendritic cells are like intelligence specialists. They collect pieces of the invader at the site of the battle. Then they travel to the nearest lymph node, triggering a cascade of events. Inside the lymph node, the dendritic cell presents pieces of the invader to T cells, the immune system's communication specialist. In turn, T cells activate B cells, training them to form antibodies, the molecular weapons that target the invader specifically. The immune system now mounts its ultimate defense. B cells release millions of antibodies, each antibody's shape tailored to identify and neutralize the enemy. The immune system deploys these tailored weapons until the infection is wiped out, ready to respond even faster the next time.
Okay, folks. So, do you have any questions about the video itself that was just shown to you? Okay. So, we're going to be talking about these processes moving forward for the next half hour or so. And, if I talk about a macrophage or a T lymphocyte or a B lymphocyte, Now you have some visualization in your mind of what these things kind of look like, okay? Uh, Early in the video, when it transitioned from looking at that guy that was playing basketball, and you went in and you started looking at things happening in the body, did you notice there was like this red cell, and then this kind of really weird arm came out and grabbed it and took it inside? You know what I'm talking about? This weird arm came out and grabbed something and brought it inside. Okay, that's what you're seeing on page 241 of your textbook. Page 241 of your textbook is an example of a macrophage going out and grabbing onto something and bringing it inside, right? That's much more exciting than the picture you see on page 241, because that's really what's going to happen, okay? But unfortunately, you know, life is not a cartoon. We got to talk about some fairly complicated ideas, okay? Any questions before I go back? Well, really what I'm going to do is start making a drawing, okay? I'm going to encourage you to have a nice big piece of clean notebook paper available and some colored pens and pencils. So that's going to help you as we go through this, all right? So next thing we're going to do is diagram out the steps of a typical immune response, okay? So let's do that. I'm going to go back to shared screen, okay? And I'll just take note in your notes, steps in a typical immune response, okay? And refer to PowerPoint drawing on this, okay? All right, so one moment here. Let's get the chat window back up, and let's get PowerPoint up here. Okay, folks, here we go. So let's see what we can do with this. Okay, so keeping in mind the visualization that you saw in the cartoon, let's do this, okay? And bear with me while I get this thing set up here, okay? Steps in the immune response. Okay. Over on the side, there's going to be this question that we have to keep asking ourselves. Is the immune system on? Okay. Is the immune system on? Let's carve out a space so that we can answer that. All right. Okay, folks. So, where to start? Okay. So, what we have here is a scenario where the first step in an immune response is that something foreign has to enter the body. Okay, something foreign enters the body. Now, you know, it could be a bacterium that gives you strep throat. It could be a bacterium that gives you tuberculosis. It could be a cold virus, which gives you the sniffles. It could be HIV. It could be hepatitis, which damages your liver. It could be coronavirus. Okay, so for right now, let's just say that we have a virus that has entered the body. Okay, we'll just say a virus. We won't say what kind of virus right now, doesn't matter. Something foreign enters the body like a virus. Okay, so make yourself a fairly large cell, Okay. And we're going to call this cell a macrophage. Okay, but your video, okay, the video you saw did show you macrophages. It also showed you other cells called dendritic cells. The principles that we're going to talk to you about here are universally true for macrophages and dendritic cells. Okay. And so let's say we have this virus, which obviously it's not drawn to scale. Okay. Okay. So something foreign enters the body, like this virus. The virus is floating around in your body fluids, and the macrophage is walking around, not literally walking, but it's floating around like a beach ball in your body fluid, your blood plasma, your interstitial fluid, cerebrospinal fluid, lymphatic fluid, all that kind of good stuff. It's floating around, and occasionally it bumps up into this virus, and it says, whoa, 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 I do not really recognize this thing. This thing seems foreign. So my question to you coming through the chat window is this, okay? In the chat window. If the macrophage bumps into the virus, how will the macrophage determine if the virus belongs in the body? Okay, that would be something you should be able of answering, should be capable of answering if you've had this content before and or if you've read this chapter before class today. If the macrophage bumps into the virus, how will the macrophage determine if the virus belongs in the body? Any thoughts about that? Okay, folks, let me give you some hints here. Okay, the macrophage is specifically going to look for a special kind of molecule on the presence of the virus that, since the virus is not naturally part of our own body, it's not going to find this molecule. This molecule has a big long name, and it's abbreviated with 3 letters. What is the macrophage? Okay, Matt, thank you for answering. It's not DNA, but that was a good guess. Something else. Okay, Emily, there we go. MHC. Folks, MHC stands for a big long phrase. Can you type it out into the chat window? Okay, so here's what it is. Thank you, Emily. All right, major histocompatibility complex. All right, folks, here's the deal. Most cells of your body, with a few exceptions, most cells of your body have special molecules on their surface of their plasma membrane that are described or named as major histocompatibility complex markers. These special molecules help your body's defense cells recognize whether something belongs in the body or not. Okay, so your cells are going to have this substance called MHC. So when a macrophage bumps into your cells and detects the MHC, the macrophage is not going to be alarmed in any way because that cell belongs in your body. But when the macrophage bumps up against something foreign like a bacterium or virus, and it does not identify MHC molecules, then the macrophage gets alarmed that maybe there's something wrong here that we need to worry with. Okay? It's kind of like a macrophage being a police officer goes up and frisks the virus, and if the police officer does not find any identification on the person he or she's frisking, then the police officer gets concerned that there's something wrong. Okay? All right. So number 2 here in our notes. Just one moment. Okay. Macrophage scans the virus for MHC. Okay. So let's go up here with this question, is the immune system on? So my number 1 here corresponds to my number 1 statement here. When something foreign enters the body, is the immune system on? Okay, at step 1, when something foreign enters the body, is the immune system on? What do you think, folks? The question is really this: when something foreign enters the body, is your adaptive immune system immediately on and responding to that substance? Okay. All right, good. The answer is, in fact, no. That's correct, okay? Because if you think about it, we have not recognized the enemy yet. Okay, number 2. Statement number 2 down below is the macrophage scans the virus for the MHC. Okay, in our conversation, no MHC is discovered, no MHC found. So if the macrophage scans the virus for MHC and no MHC is found, is the immune system activated at that point? At step 2, is the immune system going to be turned on? Okay, Quinn says yes. Do you agree with her? Alyssa says yes. Catherine? Okay. All right, thank you for participating. The answer is still no. It is not. Okay, it is not. Okay, now this is a nuance. Here's where the philosophy becomes. Okay, so let me stop sharing and talk to you. Okay, remember a couple things. Macrophages are second-line defense cells. They are not third-line defense cells. They are second-line defense cells. Okay? Now let me ask you a question. If we were all sitting in a classroom together, if we had been able to have a normal class and we were sitting in a classroom together for 8 weeks, we would get used to each other. We would get used to seeing each other. If today, as we are all in the same room together, someone logs into our virtual classroom that has not been with us before, and you looked at that person, and you looked at that person in the room, and you haven't seen that person before, you would know that that person is new. You would know that that's unusual. That person's not normally found. But if we're in the same room together and someone new comes into the room and sits down, simply by looking at that person, are you going to know that person's name? Do you see my point? If someone comes into the room that we are not used to looking at for 8 weeks, and they come into the room for the first time today, you would say, oh, that's a new person. But just looking at him or her is not enough information to know that person's name. When we say that the adaptive immune system is able to do what it needs to do, it means that the defense cells, the B and T lymphocytes, needs to know the name of the pathogen. We don't know the name of this pathogen right now. All the macrophage has done is bump into the pathogen and look for MHC to determine if the virus belongs to the body. Belonging in the body is not the same thing as knowing the identity of the pathogen. Does that make sense? Let me say it again. Knowing that something is foreign in the body is different from knowing the name of the foreign thing in the body. Okay? Questions? All right, let's continue. All right, so you should be seeing the PowerPoint drawing again. So the macrophage scans the virus. It does not find any MHC. The immune system is still not on because we don't know the name of the virus. We know that the virus doesn't belong in some generalized sense, which creates a little bit of alarm in your body, but it's not the same thing as recognizing the name of the foreign material so we can create a proper adaptive immune response. Okay, so number 3 here. Since the macrophage did not find any MHC, the macrophage is going to get alarmed. It's kind of like a police officer not finding any identification on a suspect. And so the macrophage, like a police officer, might arrest the suspect, but that's really not what the macrophage is going to do. The macrophage is going to ingest the suspect. The macrophage is going to consume the virus through phagocytosis. So this virus is going to literally be brought into the cell. Okay, through the process of phagocytosis. All right. Okay, gang. So the macrophage does not find any MHC, so the macrophage eats the virus. Macrophage eats the virus. And in step 3, the answer to that question is still no. Okay, we have not turned on the immune system yet. So what's going to happen next is that the macrophage is going to take the virus inside and it's going to destroy the virus. Okay, folks, through the chat window, type your answer in. Okay, what organelle of the cell did we say is often used for defense? What organelle of the cell is often used for defense? Do you remember? See, this kind of gets to your question, Quinn, about the final exam. Am I going to explicitly go out of my way to ask you questions from other chapters? No, but I still need and expect you to know basic knowledge that you should have learned already. Okay, all right. All right, so yes, what I'm fishing for here is the lysosome. because, you know, the lysosome is inside the cell, which is right where the virus is at this point, right? So we have this lysosome here. All right. And I can't draw too well, so I'm not going to even try to draw it. But suffice it to say, like putting a branch of a tree through a wood chipper, the virus is going to get passed through the lysosome and spit out the other side, so that what gets spit out are various fragments of the virus. So these are viral fragments. Okay. Okay. So this virus has effectively been destroyed. It's gone. And you might say to yourself, well, okay, this virus has been destroyed, so there's really no further threat to the body. But think about it. If you are infected with a virus, you're not going to be infected with a single virus particle. You're going to be infected with 50 million virus particles, right? If you're sitting in a bus next to somebody that has a cold virus, or God forbid, coronavirus right now, and they sneeze or cough, right, they're going to emit millions and millions of virus particles that you may inhale. So my point is, even though we have now killed this single virus, there's still millions of other clones of this virus floating through the body fluid. So it would be improper to lower our defenses. So long story short, here's what's going to happen next. What this macrophage is going to do is it's going to take its rough endoplasmic reticulum, and it's going to use the ribosomes that are embedded on that membrane to manufacture MHC proteins. Okay, so the rough endoplasmic reticulum inside this macrophage is going to decode pieces of messenger RNA that the cell makes from its genes, its DNA, to manufacture MHC proteins. Okay? And what the cell is going to do is it's going to take the viral fragments that got chewed up by going through the lysosome, and it's going to take these viral fragments, and it's going to bring them down, and it's going to glue them onto the end of these MHC proteins. Okay? So I guess down here we could put number 4. Viral fragments glued to MHC. Okay. And then does anybody know what's going to happen next? Does anybody know what's going to happen next? The answer to our question up here for step 4 is still no. Okay, so what we have is that the cell has destroyed the virus. It has made some MHC proteins, and it is gluing the viral fragments to the MHC proteins. So what happens next? What happens next, folks? Can anybody state what will happen next? Again, if you've dug into the reading of this chapter, I think you should be able to answer this question. Okay, Let me help you out, folks, because we got to move forward because I definitely need to get to chapter 14 today. Gang, here's what's going to happen. So these fragments of virus, Quinn, you asked, do T cells start killing the virus because they don't have the MHC? The answer to that, Quinn, is no. The reason it's no is because we have not turned the immune system on yet. The T cells are not even aware of the virus at this moment in time. Okay? Any other questions? All right, so gang, here's what's going to happen next. This cargo, which I'm going to draw, which I'm going to circle in sort of a purplish color here, this cargo is going to be moved to the surface of the cell and placed outside the surface of the cell so that I now have the MHC with the viral proteins stuck to it. Okay, forgive me for being slow at this. Okay, this material is put out on the surface of the cell. Okay, so the next step is that this cargo of viral fragments that have been glued to the MHC proteins are stuck outside the cell. Okay, question coming through the chat window. What process is this macrophage performing when it puts viral fragments outside the plasma membrane? What process is this macrophage performing when it puts viral fragments outside the plasma membrane? Okay. I realize it's extremely easy for me to say, oh, you should be able to answer that if you read your book. You should be able to. But I understand that learning is a hard process and you often have to cover the content multiple times before it really starts to stick. So can anybody answer this question? Okay, let's move. I'll move it along. I will answer it for you. The answer to the question is a process known as antigen presentation. Antigen presentation. Okay, when the viral fragments are placed outside the cell is doing something called antigen presentation. And in fact, the macrophage is now known as an antigen presenting cell. Okay. So let me come up here because I'm going to run out of space pretty quick. Number 5. Okay. Material placed outside the cell. The cell is now an antigen presenting cell. Okay, antigen presenting cell. All right, folks. So the answer here for number 5 to this question, is the immune system on, is no, 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 no. So this material is placed outside the cell. The macrophage is now an antigen presenting cell. Okay, do you have any questions? Okay, here's a question for you then. Coming through the chat window. Why does the macrophage put viral fragments outside the cell? What is the purpose of this? Why does the macrophage put viral fragments outside the cell? What is the purpose of this? Okay, Alyssa, to dispose of waste. That's a nice idea, and it's a very pragmatic one. And I wouldn't necessarily say no to it, but that's definitely not the answer, but I thank you for trying. So again, why does the macrophage put viral fragments outside the cell? Notice that we have not answered yes to the immune system being on yet. We haven't answered yes. So there's something here that's tying this together. What is the purpose of the macrophage placing digested viral fragments out on its surface? What's the purpose? Okay, Quinn, the purpose is to turn on the immune system. That's true. So let's take this the whole way to completion. Quinn and everybody, what is this macrophage currently trying to attract? What is this macrophage trying to attract? Okay, you say T and B cells. That's a good answer. I like it. And let me narrow this down. Folks, which kind of cell is it? The T cell or the B cell that's capable of turning on the immune system? Okay, Nick, Catherine, Matt, very nicely done. Okay, gang, we got it. So the answer to my question, the question is, why does the macrophage put viral fragments outside the cell? The whole reason this is done is to attract the attention of a T cell, specifically a helper T cell that has the correctly shaped receptor capable of binding one of these digested viral fragments. Okay, so let me type that into the chat window to answer this question. The antigen presenting cell is trying to attract a helper T cell with the correct receptor to bind to one of these presented fragments. Okay, all right. So what we need is for a helper T cell, and I won't be able to draw it to scale, but I'll just draw it here. We need a helper T cell. Okay, so T subscript H, a helper T cell, that has a receptor capable of binding to one of these fragments. So I'll draw it like this, okay? You might imagine that if we had a receptor that looked kind of like a square box, it could probably dock inside this tuning fork receptor, okay? That's what we need to happen, okay? So step 6. A helper T cell with the correct receptor binds to the presented material. Okay, please note, folks, that if step 6 happens correctly, then the immune system actually turns on. Okay, this is the binding event. Remember I said a while ago we needed a binding event to actually turn on the adaptive immune system? Well, right here is the binding event that I'm talking about. This is the binding event that will lead to immune system activation. Okay. Please note that if step 6 does not happen for a variety of reasons, if step 6 does not happen, the adaptive immune response never starts. It never turns on. Yes, the first and second lines of defense will continue to operate, but the third line of defense, the immune system, the adaptive immune system, will not turn on. Okay, and the outcome of that could be that you might get overwhelmed by the infection and pass away from it. Okay. Gang, do you have any questions at this time? Okay, so I'm going to come back to this, but I'm going to stop sharing to ask you some questions. All right. So folks, at this moment in our note taking, we have a helper T cell that is bound to a fragment on the surface of the antigen presenting cell. Okay. So, folks, my question to you is this. Why should I expect the body is capable of having a helper T cell bind to this presented material? Why should I expect that that event can actually occur? Once again, why is it reasonable to expect that your body will have a helper T cell capable of binding these viral fragments? Why should I expect that that can happen? Okay, Quinn. Quinn's the first one to answer, and she's right. There are so many T cell receptor combinations, because remember we talked about how can you be sure that you win the Powerball lottery? You buy all the number combinations for all the tickets. Well, before your body was infected with this virus that's in your notes, your body was making T cells and putting randomly shaped receptors on their surface so that this binding event has a good probability of actually occurring. Okay? All right. Do you have any questions right now? All right. All right, folks, so I'm looking at my time, getting a little bit worried about it, so I'm going to move this conversation along. You need to let me know what you don't understand, okay? All right, let's continue here. And we're back. Okay. All right, so at this point, if step number 6 has occurred correctly, the immune system will in fact turn on. Okay. Now I'm going to go to a second screen in the PowerPoint drawing. Okay. What's going to happen next is this T cell is going to release a bunch of molecules called cytokines. They were referenced in the video I showed you. Cytokines are signaling molecules. Just like hormones are signaling molecules in the endocrine system, cytokines are signaling molecules in the immune system. But we're not going to have time to go into detail, but suffice it to say, this binding event is going to cause this T cell to release cytokines, which activates other B and T cells to get busy fighting this specific pathogen, this specific virus in this case. Okay? So let me just show this as so after this binding event has occurred, this T cell is going to release a bunch of molecules, which are cytokines. The cytokines are going to activate the rest of the B and T cells that are capable of fighting this specific virus. Okay? So let me insert a new slide here. Okay. All right. So step 7 is going to take place in two different, step 7 is going to happen simultaneously. Okay. And it's going to happen as 7A and as 7B. Okay. And here's where, now that the immune system has been activated, you're going to have clonal selection occur. Okay, clonal selection is a process that is talked about in your textbook. You're going to have to spend some time thinking about these things. Figure 1310 on page 248. Okay. Figure 1310. Clonal selection is going to happen for the B cells. And clonal selection is going to happen for the T cells. Okay. And what I mean by clonal selection is the body is going to precisely turn on those B and T cells that have the correct receptor to recognize this specific virus. Okay, clonal selection means you have identified the specific member of your defense army that has the capability of binding to the specific threat. And what you're going to do is take these specific cells and you're going to amplify them by reproducing and copying them many times. In other words, you're going to create a clone army of B cells. Okay, so I'm referencing, in some ways, I'm referencing Star Wars. Okay, so folks, in the original Star Wars timeline, if you're familiar with Star Wars, what's true about all those guys underneath the white paratrooper outfits? What's true about them? In the Clone Wars of Star Wars, what's true about the white paratrooper guys? Do we have no Star Wars fans out there? Okay, Matt, they're not too good at their aiming. Very good. I like it. They are all the same size. Yeah, Matt, they are all clones of Jango Fett. That's right. Okay, in the original Star Wars mythology, all the dudes inside the white paratroopers, if you were to take their helmets off, they would look exactly the same because they were all clones of one individual. Okay? So, when we have clonal selection for B cells, we're going to make a clone army just like a Jango Fett army in Star Wars. We're going to make a whole army of B cells that have the right receptor to recognize the pathogen. And we're going to make a whole army of T cells that have the right receptor to bind to the pathogen. Okay? Because let's face it, if you have one cell that has the right receptor to bind the virus, that's fantastic. But how can that single cell possibly defend the body against 50 million virus particles? It cannot. It needs help. So to get help, the body takes the cell that it identified in step 6 and reproduces it millions and millions and millions of times to make a clone army of T cells that have the right receptor to bind this specific pathogen. Folks, is this making sense? What questions do you have right now? Okay. The immune system has activated because it discovered a T cell that could recognize the pathogen. But that single T cell is not capable of fighting all 50 million virus particles. So it has to activate other T and B cells that can also recognize the pathogen, and it has to order them to reproduce and multiply millions and millions of times to create clone armies of the B and T cells that can all fight the 50 million virus particles. Okay? Questions? Okay, so I know this is fast, and folks, let me tell you, the immune system is an amazing, amazing thing. It's also an incredibly deeply complicated topic. Okay, you could easily spend a full year taking a proper immunology course. Okay, so the clone army has been built, and that's going to lead to antibody-mediated immunity. And it's going to lead on the T cell side to cell-mediated immunity. Okay, I want you to see that I am talking about things from your own textbook. Okay, so, right, the cell-mediated immunity is on page 251, figure 1313. All right, and the antibody-mediated immunity is on page 249, figure 1311. Right, I mean, this is pretty deep stuff, even for a beginner course. It has to be, because to even get a basic understanding of the immune system, you have to dig in a little bit and do a little bit of sweating. Okay, so that's what I'm talking about at this moment. Okay. All right, so for the antibody-mediated immunity, the B cells that have been activated, they're going to reproduce and become plasma cells. All right. If you look carefully at page 249, page 249, identify the plasma cells. Somebody please tell me, what kind of material do plasma cells manufacture? What material do plasma cells manufacture? Okay, page 249. Okay, thank you, Nick and Alyssa, Emily, thank you very much. That's exactly what I'm after. I'm pointing out, folks, that your antibodies are made from the plasma cells. The plasma cells get made by activated B cells that are part of the clone army. So plasma cells are a subcategory of activated B lymphocytes. Okay, so the plasma cells, they make your antibodies. Okay. And if I can get my technology to work, I will write that, but just a second. Have a little difficulty here. Come on. Okay, so the plasma cells make the antibodies, and I'm going to have to leave it to you to spend your time understanding what antibodies are and kind of what they do for you. Obviously, you're going to want to look at figure 1312. There could be pictures, there could be questions about antibodies on your exam, and also table 13.4 on page 250. So the plasma cells make your antibodies, and antibodies, generally speaking, will go out and they will bind to the surface of pathogens, the surface of viruses and things like that, and act as a marker for other defense cells to come over and attack whatever the antibody has attached itself to. At the same time, the other populations of B cells are not going to become plasma cells, but they're going to become memory B cells. Memory B cells live for a long time. Now, long is a relative term, right? Some cells of your body might live a week, other cells of your body might live years. Memory B cells, on average, will live for about 10 years, give or take a little bit. So they live for about plus or minus 10 years, you know, a little bit longer, a little bit less. Okay. And the whole idea of the memory B cell is exactly what you think. The memory B cells, their purpose is that should you become infected with the exact same pathogen 5 years down the road, these memory B cells will recognize that pathogen. You will jumpstart your immune system much more rapidly than you did the first time, and you will clear the infection from the body. Okay? It's kind of like having trained police officers who have experience with this specific crime boss, if you will. All right, so moving things forward, on the T cell side, the cell-mediated side, this is going to be carried out by your activated T cells. These T cells are going to also reproduce and specialize. And what I need to highlight here for you is that they're going to become two different kinds of cells. They're going to become effector. So I'm looking at page 251 of your textbook. Effector, cytotoxic T cells. And somebody tell me, what kind of substance is the effector cytotoxic T cell releasing? What kind of substance is the effector cytotoxic T cell releasing? Have a look at figure 1313 on page 251. What substance is the effector cytotoxic T cell releasing? Okay, beautiful, Alyssa and Quinn, exactly, perforin. Take a look at that. Perforin, which is in cartoon bubble 6B of figure 1313. Folks, perforin refers to special molecules that have the ability to form holes in the target. By forming holes, we then can have too much water enter or exit that cell and destroy that cell. So the T cell is kind of like the assassin that goes out and releases these toxic substances that will then destroy virally infected cells. Or if we're talking about a bacterium, it might go up and attack the bacterium directly and kill it through the release of perforin and other kinds of molecules. Okay? So effector cytotoxic T cells will release perforin. Sorry, folks, my tech is not working good today. Having trouble with this. So I better finish this up here. Okay. All right, so perforin is released from effector cytotoxic T cells. And then we also have memory cytotoxic T cells, which are created. Okay, so both the B cell antibody-mediated branch of the adaptive immune system and the T cell, the cell-mediated immunity branch of the adaptive immune system, will develop memory cells, which will live a long time, give or take 10 years plus or minus a few years, okay? And they will respond to the same pathogen quickly the second time around. When you get a vaccine, your body will go through this process and develop these memory B and T cells. But you may know that sometimes you need to get a booster shot, right? Think about the last time you had a tetanus shot. It might have been a long time, and if it's been a long time, you might need a booster tetanus shot, because eventually these memory cells do pass away and you need to bolster your immune system again against the same pathogen. Okay. Folks, there's still a whole bunch more that I could tell you, but I'm going to let it rest at this point, because at the very least, I think I've given you the support you need for your final exam. So, let me ask you, do you have any questions about the immune system before we start chapter 14? Any questions before we start chapter 14? For you younger folks, I think you're going to live in a time of medicine that's going to be truly amazing, as long as we don't destroy ourselves with the societal problems we currently have. But future therapies for a lot of diseases, including cancers, are going to be immune system based. As we gain greater ability to control these cells in our body, we're going to be able to do some amazing things. Quinn, can an illness kill T cells? Yes, that's exactly what HIV does. Think about it. HIV is human immunodeficiency virus. Immunodeficiency means that in the case of HIV, T cells are killed. And if the T cells are killed, you cannot activate the adaptive immune system, which means something like a strep throat bacterium could then be deadly to you. Other questions? All right. Folks, I would encourage you, if you're interested in biology and at some moment in time you have a chance to take a full course in immunology, that you do it. It will be hard, but it will be very rewarding too. It's fascinating stuff. Okay, gang, I'm going to go back to the chat window, back to the share window. We're going to start chapter 14. Okay. So I will send you that little cartoon, but hopefully, you're all capable of drawing better than I am. Hopefully you have your own copy of it at this point. All right, so chapter 14 is the respiratory system. Nothing like covering that in 40 minutes. I'm being sarcastic there, right? Because everything we've talked about this semester is just pretty deep. At any rate, we're going to try to give you support for your exam. Let's talk about the respiratory system. Respiratory system as a topic begins on page 268 of your textbook. Okay. So question, let's start the chapter with a question. Okay. What is the primary function of the respiratory system? What would you say the primary function of the respiratory system is? I'm also gotta get the chat window up here. Okay, chat window is back up. What is the primary function of the respiratory system? Okay, so Quinn, you say to put oxygen in your blood. Well, that gets us a pretty long way to the full answer. Okay, Nick, you say to aid in cellular respiration. I like that. I like that. And that brings in some complexities, but I definitely agree with you. Okay, so what I'm going to put down in your notes is that the primary function of the respiratory system is to provide gas exchange between your blood and the tissues of your body, between the blood system and the tissue of the heart, between the blood system and the tissue of the brain, between blood and the tissue of your kidneys. That's the primary function. Now, of course, we think about oxygen because we need to deliver fresh oxygen to the cells of our body to support that aerobic cellular respiration. But remember, gas exchange is a two-way street. Not only do we got to put oxygen into tissue, we got to get rid of carbon dioxide as well. Okay. Now, when we talk about the term respiration, that's a little bit of a loaded word because it's very contextual. What does respiration refer to? Well, it can refer to cellular respiration, which Nick, you answered in the chat window, and I'm happy you did that. Okay, and that was the topic we covered briefly in chapter 3 a long time ago. And it can refer to what's known as organismal respiration. Okay, both of these topics are definitely intimately related. However, that being said, I'm really not going to focus on cell respiration because you had at least some exposure to that in chapter 3. What I'm going to talk about is the organismal respiration component of respiration. Okay, so let's define it this way. Organismal respiration has 4 components. Okay, let's just push that down. Okay, organismal respiration has 4 components. They are, okay, now when you say breathing, that's kind of a slang form for what you're really doing, which is ventilation. Okay, so when you say breathing, you're really talking about ventilating. Okay, and by that, what I mean is moving oxygen-rich air into the lungs and moving CO2-rich air out of the lungs. Okay. Now, if this part by itself is not sufficient, right? Because moving the gas into the lungs or pushing the gas out of the lungs does not do anything to get the gas into or out of the bloodstream, right? So this by itself is not enough to complete respiration, but it obviously is an important component. And if I'm being frank, if you've seen the study guide, the ventilation here that I'm talking about is really the topic of your essay question, okay? So ventilation is mechanical in nature, okay? And we're going to be talking about that in just a few moments. The second component of organismal respiration is what is referred to as external respiration. And by external respiration, what we mean is the exchange of oxygen and CO2 gases between the lungs and the bloodstream. Okay, so the oxygen that's in the lungs must cross the respiratory membrane and the alveolar sacs of your lung tissue to get into the bloodstream, and the CO2 that's in the bloodstream must go in the opposite direction to get into the lung anatomy so it can eventually be exhaled. The third component of cellular respiration talks about gas transport. Okay, by the transport of gas, I mean the transport of the oxygen in the red blood cells as the heart contracts and pushes blood along. But how is the blood holding on to the gas, right? When we talk about the motion of the blood is handled by the cardiovascular system. But how gas is moved within the blood is the domain of the respiratory system. Okay? So this deals with how the body moves gases through the bloodstream. Number 4 is internal respiration. Okay? Internal respiration is the exchange of oxygen and CO2 between the bloodstream and whatever target tissue the blood is at, whatever capillary bed we're talking about, right? If we're at a capillary bed at the small intestine, then that's where the internal respiration is going to be. It's going to be between the blood in the capillary bed and the surrounding cells of the small intestine. We're going to push oxygen into those cells. We're going to let CO2 come out of those cells and get back into the blood. All right. So these 4 components together make up what's known as organismal respiration. First, you got to mechanically move the gases into the lungs or out of the lungs. Then you got to get those gases into or out of the bloodstream that's at the lungs. Then we got to have those gases bound in some way, typically by hemoglobin in the red blood cells, so that as the heart moves the blood, the gas can be moved along with it. Then once that blood reaches its destination, you need another exchange of gases to get the oxygen into the proper tissue and CO2 out of that tissue to be removed so it doesn't poison the tissue. Okay? Okay, so that opens up. Do you have questions at this moment? Obviously, after internal respiration has occurred, the oxygen that has been injected into the small intestine tissue will enable those cells of the small intestine to use that oxygen for aerobic cellular respiration purposes to make ATP. Okay. So maybe we just put a little, this supports aerobic cellular respiration. Okay. So question coming here into your notes. Let's see. Why is it dangerous for CO2 to remain in the body? Why is it dangerous for CO2 to remain in the body? Right? We just got done outlining organismal respiration and the idea that eventually the CO2 has to come out of the target cells, get into the blood, be transported back to the lungs, where the lungs can then exhale it out of the body. But that process doesn't work efficiently, or it doesn't work correctly, and CO2 levels start to rise in the body fluids, particularly the bloodstream. How and in what way is that dangerous? Okay, Quinn. So you say it prevents oxygen from attaching to the hemoglobin and we suffocate. There is a little bit of truth to what you are saying, but I do believe you might be confusing your answer with carbon monoxide. Okay, carbon monoxide will do what you're saying, and very, very high levels of CO2 could also do the same thing. So there is some truth in what you're saying, but that's not the primary threat here. What else would be dangerous here, folks? Okay, folks, let me ask you this question. I'm going to ask another question through the chat window. Why do scientists warn us that excess CO2 in our oceans is dangerous? Right? Everybody's worried about global warming at this time for a variety of reasons. It's been a hot topic for a long time. Why do scientists argue that excess carbon dioxide in our oceans is a bad thing? Okay, Matt, you say it depletes the oxygen level. Definitely it will lead to a shift in the oxygen level. That's true, but there's something else here that I'm fishing for. Okay. Does it have to do with heat? It does not. Okay, I think I better help you folks with this. Folks, here's where a background in chemistry would help you. Okay. CO2 is a substance that will combine with water and act as a substance that will form an acid. Okay. CO2 combines with the water of your blood plasma to form an acid called carbonic acid. Okay. Now I'm not gonna hold you responsible for some chemistry and things like that. Okay, but take a look at page 277. Page 277 in your book. Okay, you will see on the right side there, you will see a chemical reaction. And if you look in the middle of that chemical reaction, you will see carbonic acid. Okay. So note page 277 of your textbook. Suffice it to say, what I'm getting at is that if you have elevated levels of CO2 in the body, for whatever reason, you will actually form an acid. You will begin to acidify your body fluids. In effect, you begin to acidify your body fluids, which damages tissue. Okay? Now, I'm not talking about the horror movies where somebody throws hydrochloric acid on someone and their face melts away. I'm not talking about that. But I am saying that you will slowly begin to acidify your blood plasma, your cerebrospinal fluid, your interstitial fluid, your lymphatic fluid. And you know from our conversations in this course that if we change the pH of an environment like your body, any molecule in that environment will change its shape and it will not function properly anymore. Okay? So that's why excess CO2 is actually dangerous to us. Okay, questions, folks? All right. Now, normally Normally I would spend a little bit more time talking about respiratory anatomy, because you should be familiar with it on page 270. Looking at the clock, though, I see we're getting short on time. So rather than going through things blow by blow and defining everything for you, I'm going to leave you do that for yourself. But I am going to take you on a very quick journey where we take a look at the various anatomy of your body that air moves through and across as it enters your body and gets to your lungs, okay? So let's do a quick flow chart of air motion through the body. All right, obviously you can inhale air into your mouth, you could be a mouth breather. And we have all been mouth breathers at times when we have allergies or colds and our noses are stuffed up, that kind of thing. But air is, Mother Nature did not intend us to breathe air into our mouth all the time for obvious reasons, including the fact that it dries out our mucous membranes in our mouth, which increases the likelihood of infection. Okay, we're supposed to breathe air into our nose, right? So starting on page 270, just look at these structures as I outline them for you. As you inhale, right, you bring air into your nasal cavities. Okay, basically your nose gang. That air then moves down into the pharynx. Okay. That air, so put an eyeball on where the pharynx actually is positioned. That air from the pharynx then moves down into your larynx. Number 3 is the larynx. Right? If the larynx is open, air moves in the correct direction. If the larynx is closed, then that means we're actually not moving air, we're moving food or drink or something like that. And in that case, we need the larynx to be closed to divert the material into the esophagus. Food, drink, moved into esophagus, okay? So if the larynx is closed, we're moving food and drink into the esophagus so it can go down into the stomach. But if the larynx is open, as it will be for breathing of normal, healthy air, okay, then that air is going to move into the trachea. From here, the air is going to move into your bronchi. From here, the air is going to move into your bronchioles, which, Many people know about bronchi and bronchioles if they suffer from asthma, because it's something that really concerns you, right? If you have asthma or other breathing disorders. Okay. From here, the air is going to move into the alveoli, which are those little gas exchange chambers in your lung tissue. They kind of, on page 270, they kind of look like tiny clusters of grapes that you might buy at the supermarket. Okay, those alveoli are extremely delicate. I may talk more about them in just a moment. Okay, so this is basically the path of air moving into your body in a natural way, from the nasal cavities to the pharynx to the larynx to the trachea, bronchi, bronchioles, and then the alveoli. Okay, at this point, you will have gas exchange into the bloodstream, and gases are distributed by blood circulation. Okay. All right. So just so you might have an idea, the alveoli, number 7 there, the alveoli are in very intimate physical close contact with capillary beds in the lung tissue. You can see that in figure 14.8 on page 274. Page 274, figure 14.8, you see that. So it should not be a surprise to you that at the alveoli, we can move the gases into the blood system. Okay. Let me just mention here, gang, I think I did state it, but the alveoli are microscopic gas exchange chambers. They are very, very delicate, extremely delicate, okay? And honestly, you know, if you do happen to smoke, you are damaging your alveoli, okay? Smoking is one thing that definitely damages alveoli. Also, you know, if you were near a heavy fire, like God forbid you get trapped in a house fire and you're breathing superheated air, the superheated air can actually damage the alveoli. Okay, so there are lots of things that can damage alveoli, but the one that, you know, we typically caution folks against is smoking. Okay, smoking damages alveoli. Okay. So, folks, how many of you, by show of hands or by talking into the chat window here, how many of you have been to a museum exhibit called Body Worlds? Do any of you know what I'm talking about by Body Worlds? Okay. All right. So before the end of summer, or maybe as a treat for yourself this fall, Google Body Worlds. Okay. Google Body Worlds, and hopefully you won't find anything pornographic, but hopefully what you will find is the museum exhibit Body Worlds. Body Worlds is a museum exhibit. There's different versions of it, where this company that was originally founded by a medical doctor in Germany takes people's bodies that have donated them to medical science and preserves them in ways that then you can go look at the museum to see the amazing intricate anatomy inside the human body. There's also one for animals, like at a zoo exhibit. At any rate, suffice it to say, I've been to 2 of them, and they are amazing things. And one of them that I was at showed an example of lung tissue from a person that had passed away from lung cancer, and you could see the lung tissue that was damaged by the smoking. The nice healthy pinkness of the tissue was replaced by a dark black material. Okay, so it is no joke when you smoke, you damage your air exchange capacity. As the alveoli get damaged, they literally collapse. It's like taking a grape and pressing it till it collapses. And then that alveoli cannot participate in moving oxygen into the bloodstream anymore. If you smoke and you stop smoking, your lungs will heal themselves, but it will take years for that to occur. So if you smoke, please stop. You're wasting your money and you're destroying your health and you're damaging the health of people that live with you. Okay? There is no positive to smoking. No positive. Do you have any questions right now? All right, gang. Let's keep going here. So I would recommend the Body Worlds to you. It can be a little bit expensive, but if you're only going to do it once in your life, it's worth the investment. And usually kids love it because it's full of all kinds of gross stuff. All right. Okay, gang. We need to talk about ventilation so I can give you some support about what's going to be tested on the exam. Okay? So let's talk about ventilation. Okay. As I said, ventilation means moving air into and out of the lungs, the mechanical nature of this. Okay. Now, if you think about it, and please, I'm going to ask you to maybe give me another 5 minutes. I don't know how quickly I can do this, but I'll get finished as quickly as I can. A couple points here, okay? Your ability to ventilate depends on the ability of your body to form or create pressure gradients, okay? By a gradient, I mean an area where you have high air pressure versus an area where you have lower air pressure. Okay. If you think about our course, we have talked about things that often go from areas of intensity or concentration, high concentration to low concentration. As an example, when we talked about simple diffusion, you remember that? Simple diffusion was the movement of a solute from an area of high concentration to an area of low concentration. So the solute went from a high concentration to a low concentration. Think about sound waves and sound energy. If you're playing your stereo at the house, doesn't it sound loudest, the most intense when you are close to the loudspeaker? And as you walk away, the sound waves are less intense. So sound spreads out from its source. Same way with heat. If you have a bonfire outside and you're close to the bonfire, you feel the intensity of the heat. If you walk away from it, it doesn't feel as intense. So heat spreads out from the source. What I'm trying to get you to see is that there are many things in our universe that operate on thermodynamic principles where the energy or the material, the mass, goes from an area where it's highly concentrated to an area where it's lower concentrated. Ventilation depends on differences in air pressure from high pressure to low pressure. And in fact, air always moves naturally from an area of high pressure to an area of low pressure. Okay? That always happens. So let me put in a little table here. I think that should suffice. And I'm going to put the first column as lung pressure, middle column as environment pressure, and third column as the direction of air motion. Okay, now when I say environment in the middle column, I'm talking about the environment outside your body, right? The environment you're in, but the environment outside your body. Okay. So let's see here. Get all of these in bold. Okay. We could put numbers on these things, and I might in just a moment, but right now I'm going to talk in qualitative terms. If your lung pressure, if the air pressure in your lungs is low, meaning it's low relative or compared to another location, such as the environment pressure outside your body, meaning that the environment pressure is high, please type into the chat window, is air going to enter the body or exit the body in this scenario? If we look at lung pressure being low and environment pressure being high, is air going to enter the body or exit the body? Okay, folks. Good precaution. Okay, Quinn, thank you. That's exactly right. Air is going to enter the body, which means we are going to inhale. Air enters the body. Okay? Now, there is nothing that you and I can practically do in the real world to have a dramatic impact on the air pressure outside ourselves, right? If you're standing outside, there's nothing you can do as a human being to dramatically alter the air pressure that Mother Nature is giving us with the weather pattern at that moment of time. Yes, I guess if we wanted to go to ridiculous extremes, we could detonate a nuclear bomb and change the air pressure that way. But you know, we're not going to do that because that's insane. So what I'm saying is, our bodies cannot alter the air pressure outside themselves, but our bodies can alter the air pressure inside themselves. So atmospheric pressure is typically 760 millimeters of mercury at sea level. And for your body to lower the air pressure, which we'll describe in just a moment, it has to do some things which will lower the air pressure to 758 millimeters of mercury. You can see that even though it's only a difference of 2 millimeters of mercury, that is enough to ensure that air will enter our bodies. Okay, if we flip this around, what we have is high air pressure outside the body and low air pressure. If we have high air pressure inside the body and lower air pressure outside the body, then that's going to set up a circumstance for exhalation. We're going to exhale. Air leaves the body, okay? Now for this to happen, again, the air pressure stays the same. It's not going to change, okay? We can't do anything with it. I mean, we can't practically change it to suit our own purposes every day. But the body can do things which will pump up its inside air pressure momentarily, and typically to allow exhalation to occur, you boost it up to 762 millimeters of mercury. Okay? All right. Any questions right now? All right. Folks, bear with me. Please give me a few minutes here. What we need to do is explain, what I'm going to do is explain inhalation to you, and then I'm going to leave it for you to explain exhalation to yourself and be ready to explain both of them on your final exam on Thursday. Okay? Understanding inhalation. Okay. All right, so there's there's two major muscles that are involved with ventilation. Do you know what they are? What are the two major muscles involved with ventilation? Okay, Matt, ventilation, diaphragm is one. They are the diaphragm, and just to keep us rolling here, and the intercostal muscles of the rib cage. Okay? The diaphragm and intercostal muscles. And you can see both of those either on page 275 or even in a larger way on page 270 in your textbook. So make sure, folks, that you know what the diaphragm and intercostal muscles are. Okay? So suffice it to say that these are the muscles of ventilation. They are the muscles of breathing. And it is the contraction and relaxation of these muscles in a controlled way which allow us to inhale and exhale. Okay? So, all right. I need to talk about some things that are going to happen at the same time, but I can't describe them at the same time. So we got to take one thing after the other. Okay? During inhalation, okay, the diaphragm will contract. Okay, folks, when the diaphragm contracts, does it move up towards your head or down towards your feet? When the diaphragm contracts, does it move up towards your head or down towards your feet? Okay, Matt, you took a shot. Actually, it goes down. It goes down, folks. Okay? But thanks for trying, and I know we're rushing here. You're going to have to work this through during your study time. The diaphragm will contract. Okay? If you don't believe me that it moves down, take a look at page 275, figure 14.9, picture A. When the diaphragm contracts, the diaphragm moves down. Okay, it moves down. And the effect of moving down is to open up the volume of space where the lungs are, which is in the thoracic cavity. By the diaphragm moving down, we increase the volume of space that the lungs are in the thoracic cavity. Okay. Not only this, but, okay, believe it or not, and I know you can't tell from the pictures in your book, the diaphragm, as you look at page 275, the diaphragm is right underneath the bottom lobes of the lungs. And in fact, there is connective tissue that has stitched the lobes of the lungs on the bottom to the top surface of the diaphragm. So if the diaphragm is connected to the bottom lobes of the lung by connective tissue, if the diaphragm goes down, what's going to happen to the lobes of the lung? Are they going to go down or up? If the diaphragm goes down, what's going to happen to the bottom lobe of the lung? Is it going to go down or up? You are correct, Quinn. Good job. Okay. Okay. So let me finish this in your notes here. The diaphragm moves down, which increases the volume of space that the lungs are in the thoracic cavity. Not only this, but because the diaphragm is stitched to the bottom lobes of the lungs get pulled down. The bottom lobes of the lung get pulled down when the diaphragm moves. And basically, that's like taking a paper bag that's compressed and then pulling on the bottom of the paper bag to open the paper bag up. You're literally expanding the space inside your lungs when the diaphragms go down. I hope this logic is working for you. I know I'm moving fast. Okay, so the lungs get pulled down, which opens the space inside the lungs. Okay, the bottom lobes of the lungs get pulled down, which opens the space inside the lungs. Okay, when this happens, that means the air pressure inside the thoracic cavity and inside the lungs is going to go either up or down. What do you predict is going to happen? If I expand the space inside the lungs, what's going to happen to the air pressure in the lungs? Okay, Nick says it decreases. Okay, Alyssa, I see your comment. I'll talk to you later. Take care. It does go down. That's right. If you increase the volume of space that a gas is in, then the air pressure, the pressure of that gas in that space will go down. Okay? The air pressure inside the lungs goes down relative to air pressure outside the body. Okay? So the motion of the diaphragm causes a drop in air pressure in the lungs relative to the outside body. We have now created a gradient, haven't we? We have an area where the outside world has a high pressure and the inside world of your lungs has a lower pressure. That means air will enter the lungs. Okay? Now, at the same time that the diaphragm is doing what it's doing, the intercostal muscles are also going to contract. At the same time as the diaphragm is contracting, the intercostal muscles of the rib cage also contract. They also contract. And, you know, I would ask you during your study time to think about it. Even think about it right now as you're sitting there in your room listening to this conversation. Think about how your chest moves as you inhale and exhale. So just for a moment, for a quiet moment, just breathe and pay attention to how your chest is moving when you inhale. Hopefully you've come to the conclusion that when you inhale, your chest actually lifts upward and forward. And that motion is made possible by the contraction of the muscles of the rib cage, the intercostal muscles. So as the intercostal muscles of the rib cage contract, they lift your chest up and forward. And by lifting your chest up and forward, you expand the volume of space in that thoracic cavity, which again lowers the air pressure, which then supports motion of air into the lungs. Okay? So at the same time as the diaphragm is contracting, The intercostal muscles of the rib cage contract. This lifts your chest up and forward, which expands the volume of space in the thoracic cavity and therefore the lungs, which are positioned in the thoracic cavity. This also contributes to a lowering of the air pressure in your lungs, which allows air to enter your body. Okay? This process is reversed to exhale. Okay? I'm going to have you figure that part out for yourself. I'm not going to explain it here. It's simply the reverse. But for you to get credit on your final exam, you can't simply say reverse this process. You've got to explain what's happening during your answer for the exam. Okay? So, folks, do you have any questions right now? I know that was quite a whirlwind of stuff. And I'll be happy to help you with anything that I can right now. All right. It looks like everybody's kind of tired. So here's the deal, folks. We're not going to see each other on Thursday. There's no class on Thursday. All you have to do is take your final exam on Thursday. So I will be checking my email tonight and tomorrow. If you have a problem, if you have a question on content that I can help you with, shoot me an email and I will do my best to answer it for you before Thursday. Okay? I will try to give you a good answer through email if I can. Do you have any questions I can help you with? All right. It looks like everybody's ready to collapse, so we'll call it a quits. Folks, I just want to say thank you for being a great group this summer. We were trying to learn in a very non-traditional environment, and I think by and large you did a nice job. So enjoy the rest of your summer. I hope you get to do something fun, and I hope you come back to school in the fall and you continue your career development. You got good things ahead of you. So folks, take care, and I will see you sometime, hopefully on campus, okay? I will see you then. Take care. Take your test on Thursday.